The article we are presenting on is the metal binding affinity and structural properties of Calmodin-like protein 14 from Arabidopsis thaliana, presented by Becca and Charlotte Biochem. Calmodin-like proteins in plants are genuinely involved in the developmental process and stress responses. However, the mechanism is unknown and more research should be conducted regarding CMLs as calcium sensors. Identifying some structures and targets of calcium binding could introduce possible methods for CML as calcium sensors. The research mainly focused on an acidic protein of CML14 since it has 3 EF-hand supersecondary structures that bind calcium. The purpose of the experiment was to produce the recombinant protein CML14 and evaluate the metal binding properties to see protein stability. This would show if CML14 has the typical features of being a calcium sensor.
So, numerous methods and materials were used to study the metal binding affinity and structural properties of CML14. First, the CML14 protein had to be produced. To produce the protein, the coding sequence for CML14 was amplified through PCR. The amplified sequence was then cloned in E. coli. The coding region was checked by DNA sequencing, and the E. coli cells were transformed and allowed to express the protein. The purity of the protein was then assessed by SDS Page and found to be at least 95% pure. Next, nuclear magnetic resonance spectroscopy was used to obtain spectra in the presence and absence of 5 millimolar EGTA, magnesium chloride, and calcium chloride to evaluate metal binding and structural changes. Another method used was isothermal titration calorimetry to determine the thermodynamic parameters of CML14 when interacting with calcium and magnesium. A sample of protein was dissolved in tris, hydrochloride, and potassium chloride, titrated in the presence and absence of calcium chloride and magnesium chloride. Data was collected for calcium binding to unbound or magnesium saturated CML14. The binding affinity constant and change in enthalpy obtained were used to calculate the change in free Gibbs energy and the change in entropy using the equations delta G equals negative RT ln a K and delta G equals delta H minus T delta S. The dissociation constant Kd was then found by taking the reciprocal of the Ka value.
So next, differential scanning calorimetry experiments were conducted using a nano DSC. The CML14 sample was heated and cooled with several reagents and scanned to evaluate if the thermal transitions were reversible. A gel-sift assay was used to evaluate the electrophoretic mobility of CML14 at room temperature with incubation in reagents. An SDS page was used to separate the sample. The fluorescence spectroscopy emission of CML14 was measured to evaluate the structure of the protein. A spectra was obtained between 300 and 500 nanometers. Sucrose 12 column with size chromatography was used to find the Stokes radius of bound and unbound CML14. Stokes radius looks at the effect of size and solvent on a protein. The limited proteolysis looks at the breakdown of CML14 while bound and unbound to calcium to see if there was a difference in the structure. The protein database, ProCyte, was used for a protein domain functional analysis of CML14 sequence and also for conducting the sequence analysis and molecular modeling. The Swiss model server was used to build a relative structural model of CML14.
The results of isothermal titration calorimetry, NMR, hydrophobic ANS, and sequence analysis of CML14 provided insight to the metal binding, affinity, and structure of CML14. First, isothermal titration calorimetry evaluated the metal binding properties of CML14. The binding affinity constant Ka and the change in enthalpy delta H were calculated. Calcium binding to CML14 is a favorable exothermic process but an unfavorable change in entropy. Table 1 shows the thermodynamic parameters for the binding of calcium to CML14 in the presence or absence of magnesium. The change in entropy nearly doubles with the addition of magnesium. The Gibbs free energy change is increased from negative 6.8 kilocalories per mole to negative 5.8 kilocalories per mole with the addition of magnesium, making calcium binding less favorable but still spontaneous because of the negative delta G. Thus, the presence of magnesium influences calcium binding. The Ka values reveal magnesium decreases calcium binding affinity fivefold. NMR was obtained before and after the addition of magnesium and calcium. Saturating amounts of calcium and magnesium caused only minor conformational changes in the NMR spectra. Differential scanning calorimetry showed a 20-degree stability increase from when calcium and magnesium were bound to CML14 compared to unbound CML14, shown in the thermograms in Figure 1A, D, and E. Protein concentration did not change melting point. Proteolysis experiments show unbound CML14 is highly susceptible to being broken down into individual amino acids. The binding of calcium, however, protects CML14 against cleavage in the presence of trypsin, as shown in Figure 1B and C. ANS evaluated ligand-induced protein conformation. There was little change in ANS fluorescence when saturating amounts of magnesium and calcium were present. The electropholytic mobility, as shown in Figure 1F, has only a small change in the presence and absence of calcium. Sequence analysis revealed that EF hands 2 and 3 couldn't bind calcium due to changes in binding loop sequence. Calcium can, however, bind to EF hand 1, since there are 6 oxygen atoms to coordinate the calcium ion. Figure 2 shows calcium binding to EF hand 1 and not to EF hands 2 and 3. Figure 2 also shows the differences in amino acid sequences, leading to the differences in structure and binding abilities.
So these results indicate that CML14 is closely related to comodulins that only have the one calcium binding site. The ITC results indicate that only one binding site, which was revealed to be the first EF hand by sequence analysis. The second and third EF hand lack the appropriate structure to bind the calcium, and the magnesium does not easily bound to calcium binding sites. There are different types of sensors to bind calcium and magnesium. Based on the smaller KD for the calcium and magnesium sensor, calcium binds easier to the calcium and magnesium sites. The ITC revealed that the functional binding site, the first EF hand of CML14, is classified as a calcium-only binding site instead of a calcium-magnesium binding site indicated by the KD values. The NMR for magnesium-bound substrate is distinct from the calcium-bound NMR. The change in amino acid residues for the recombinant CML14 created smaller binding sites, which favors the magnesium binding and produced a calcium-magnesium binding site, which contradicts the previous data collected. Further NMR testing and displacement reveals consistent values with calcium and magnesium sites. As calcium competes for the binding, it decreases the calcium affinity of binding with inhibitory properties. Using the ITC data, it was concluded that the presence of magnesium decreases calcium affinity by 5-fold in CML14, which leads to an overall increase in calcium concentration in the plant cytoplasm since less is bound. The IDSC experiment concluded that the metal binding of CML14 provides thermal stability, increasing it by 20 degrees. Binding calcium to CML14 actually protects it from proteases by blocking the N-terminus. The ANS data indicated that binding calcium does not result in more hydrophobic surface. It does not induce a large conformational change, which is often seen in non-recombinant calcium sensors. The change in amino acid sequence changed the number of NMR peaks and showed a local structural change in the first EF hand. Generally, a calcium sensor protein must have the ability to structurally respond to a large range of calcium concentrations inside of the cell. The small conformational change eliminates the possibility of CML14 sensor being the traditional calcium sensor. Based on the decreased calcium affinity and the single binding site on CML14, it is unlikely that this implies a calcium binding buffering capacity. Overall, the authors concluded CML14 is neither a classical sensor nor a calcium buffer. The mechanism is unknown. Outside resources were consulted to evaluate possible functions, which indicated possible connections between CML14 and plant development and responses to stress. The CML14 genes reported activity during seed and pollen germination and during exposure to cold temperatures. The only possible discovery in terms of function was the involvement of CML14 in biotic or abiotic stimuli. Unlike other CML14 is not affected by the stimuli, whereas the other CMLs show upregulation. In the future, the hope is to identify the overall mechanism of CML14 through future studies.
In conclusion, this paper revealed advances in knowledge of structural properties and metal binding affinities of CML14. It was found that only one calcium binding site exists on CML14, and the magnesium binding to this site decreases calcium binding 5-fold. EF hands 2 and 3 were found to be non-functional concerning calcium binding. EF hand 1, however, does bind calcium. Metal binding to CML14 provides thermal stability and protects from proteases. Overall, the researchers cannot propose a function of CML14 and view it as neither a calcium sensor nor a buffer. Future research hopes to gain more understanding of the mechanisms involved with CML14.